All right, make sure I got everything here. All right, thank you everybody for joining us today. Again, my name is Ryan Gentry. I'm a sequencing specialist with Illumina. And so I want to thank the ICBR and David for having us come in today and be able to speak with you all. So when I talked with David, he had two things that he wanted me to specifically come out in front of you and talk. The first talk was going to be metagenomics, but we had a hurricane that came and kind of canceled that talk. That has been rescheduled for tentatively January 26th. So I will be back to talk about that if you're interested. But the other thing he asked me to come and speak about was cancer genomics. It's obviously an up and coming thing, and it's very important for a lot of researchers here at UF. And so with that, what we're going to talk about primarily today is multiomics and how it fits into cancer research. And what I really want to do is talk about three different things. The what, and the what's going to be basically what can you do sequencing-wise to study cancer research. The why do we specifically want to do this for cancer research? What are we looking at? What does it help us with? And then the how. The how is going to be three papers that we're going to look at specifically that kind of look over examples of using multiomics and what they found specifically using those different multiomes to better stratify patients as far as treatments, and then also possibly mechanisms for drug discovery as well. So with that, we'll probably just take questions at the end. And then I'll give the slide deck out to Chris and David. So if you want to have it, you can look over it yourselves as well. And I think they're recording this for anybody that wants to look over it again. So what can you study on the Illumina instrument? So again, we want to talk about some of the assays. You may be familiar with a lot of them, but there may be some that we just talk about that you weren't very familiar with. And so there's a lot going on the slide, but let's break it down real quickly. So the first thing you see is that we have kind of the central dogma of genomics. We have proteomics, epigenomics, genomics, and transcriptomics up there. Now we can study each one of these using genomic or using sequencing in some shape or form. Now, the way we go about it's going to be different. So the resolution down here at the bottom left-hand corner is going to be dependent on what you're trying to figure out. So if you've been to a single cell talk, what they'll use is this smoothie assay, the smoothie example, where you have a smoothie, then you have a bowl of fruit, and then you have like a fruit pie. So when we're talking about bulk transcriptomics or bulk sequencing in general, what we're doing is we're just taking all the cells, we're homogenizing, we're taking either the DNA or the RNA all together and running it sequencing. And what you're going to do is kind of get the average of everything that's happening. Now, if we take it from the single cell level, what we can do is we can individually look at each individual cell and tell what specifically mutations or what specific transcripts are happening in each one of those specific cells. And then moving on to spatial, obviously what we can do is we can look at DNA or RNA or proteomics, but we look within the specific tissue that we're looking at, and we can tell where those transcripts are happening. So if you're looking at, say, brain tissue, heart tissue, or if you're looking at cancers, you can see where things are happening within that specific slide that you're looking at. And then the last thing is liquid biopsy. So we'll go over this a little bit in more detail in the kind of fourth section that we go over. But really what we're looking at here is sometimes we're not going to be able to excise tumors out of patients. So we need other ways to be able to identify, say, biomarkers that are happening within patients to see that if there's cancers that are happening or if there's cancers that are developing within specific patients. So liquid biopsy is probably the most up and coming one of these specific assays that we're going to be speaking of today. So again, here's the central dogma, and we're going to go over each one of these individually. So first with genomics, obviously what we look for with DNA is going to be SNPs, indel, structural variants. We can look at some fusions with DNA. And so those are kind of be a lot of the hallmarks of cancer. And so what it's going to allow us to do is we can kind of tell susceptibility of cancer, predict prognosis, classify tumors, or diagnose disease. And some of the methods that we've used to do that are over here on the left-hand side. So whole exome and whole genome sequencing you might be familiar with. Now, whole exome, we just look at the exons and we need a lot less sequencing. But lately in cancer biology, we know that there are some regions that are outside the exonic regions that are relevant. So whole genome sequencing with the cost going down is becoming more popular in cancer genomics as well. And then we see circulating DNA that can be circulating tumor or circulating or cell-free DNA that we're looking at, and then CRISPR screening. If you're doing any perturbation sequencing where we're looking at any of the CRISPR changes that we're doing within any of the cells that we're specifically looking at. So the next thing is epigenomics. So with that epigenomics, what we're trying to do is we're providing a mechanistic context in how the genome is regulated in cancer. And there's a few ways that we can do that. So One of the main ways that you might be familiar with is methyl-seq. So we'll look at 5-methylcytosine or 5-hydroxymethylcytosine and basically see if some area is methylated or unmethylated to see if there's going to be possibly upregulation or downregulation in those specific genes of interest. The next thing is ATAC-seq, and this may be a little less familiar with all. So this is assay for transposase accessible chromatin. So what this uses is it uses transposase. We put that into our nuclei. And what it's going to do is it's going to chop up areas that aren't chromatin-bound DNA. And it's going to be like a counting assay. So we're just going to look at the areas that are open. And what that's going to tell us is we can look at either transcription factor binding sites, or we can look at promoter enhancer regions to give us an idea, again, of what might be happening transcriptionally or what's causing the transcriptional sequencing that we are seeing if we're doing a multi-omic type approach. And then ChIP-seq, cut and tag, and cut and run are ways of looking at kind of histone modifications. So ChIP-seq is kind of the main method, the oldest way of kind of doing it. What you would do is you would chop up your DNA sonicate your DNA, and we would add protein tags, and then we would use antibodies to pull those specific protein-bound DNAs out. We can do that with cut and tag and cut and run. And with cut and tag and cut and run, specifically what you're going to do is you're going to bind a single antibody to that DNA or that the protein. You'll bind a second antibody, and that second antibody is going to have the TN5 transposase. And instead of using sonication like ChIP-seq does, and it cuts it randomly, what it's going to do is the TN5 transposase is going to cut on the sides of that protein, so you kind of know the area specifically that you're doing. And what's great about the cut and tag and cut and run method is you need a lot less cells. You can get down to like 5,000 cells for this method. And then you need less sequencing as well. So typically you may need like 50 million reads for a ChIP-seq assay. With cut and run and cut and tag, you can get down to 8 million reads because you don't have a lot of background with those approaches. So other methods that we can use to look at epigenetic profiling with sequencing. Now, one of the mainstays obviously for cancer research is going to be transcriptomics. So this is going to connect us from gene to gene function, and it's always been necessary to assess the causes and consequences of cancer phenotypes. And so the mainstays for that is going to be RNA-seq. We can do that at a single cell or bulk RNA-seq level, and you can do that with total RNA-seq, where you're looking at long non-coding, or if we're looking at just the coding regions, we can look at an mRNA approach. And then you also have things like microRNA-seq, non-coding RNA, and cell-free RNA. And we'll specifically talk about cell-free RNA in a little bit here. And the last method is proteomics. So this is kind of linking our genotype to phenotype directly by measuring the functional consequences of genomic, epigenomic, and transcriptional regulation. And so we obviously, sequencing, well, it may not be obvious, but we can't sequence protein specifically. So what we're using is we're using antibody tags to find either proteins on the cell surface of our cell, proteins on our cell surface, or intracellularly. And so what we'll do is we'll tag those antibodies. They have a little DNA tag on them. And once they're bound, we'll wash away any other antibodies that aren't bound, and then we'll cut those tags off. And that will help us identify what proteins are present in our either cells or are in our mixture, depending on you're doing a bulk approach or a single cell approach. So a bulk approach to doing this would be Ben-seq. One of the is one of the main players. I was trying to think of the company that they used. Tapestry-seq, the Tapestry instruments by Mission Bio. You can look at DNA at the single cell level, but you can also look at proteins intracellularly that way. Site-seq is the single cell way of doing it. It's also known as AB-seq or REAP-seq. And then Olink. Olink is a company that does profiling of proteins by sequencing, but they can do thousands. So they have a couple different kits to do that. So these are kind of the mainstays and the methods that we want to do. And what we're wanting to do with specifically with this talk today is show you how we can do multi-omic approaches. So using more than one of these approaches to get to the answer that we're specifically looking for in understanding our cancers. And so why are we looking at multi-omic approaches recently? Well, before we didn't do it because it was obviously really expensive, and sequencing still is expensive, but it's getting a lot cheaper. You can see that there's a lot of companies moving into the market. And with that, that helps the consumers. It helps you all because it drives down the prices. And so you have a great sequencing core here that has some of the state-of-the-art instrumentation that gets the sequencing down about as low as you can get at this moment. And so as you can see here from 2001 versus 2022, you can do about a 30x whole genome at about $200 nowadays going into 2023. So the next thing, why use omics in cancer research, and where does Illumina sequencing fit into the study of cancer? So we no longer view cancer as a homogeneous disease. It's heterogeneous. We can look at each one of our cancers, whatever we're studying, and it can have a different mixture of all these hallmarks over here on your right-hand side. So we may have a cancer that has tumor promoting inflammation, genome instability and mutation, and resisting cell death. And then another one might be totally different, where we're looking at deregulation of cellular energetics, inducing angiogenesis, and maybe evading tumor suppressors. And so what we want to be able to do is characterize each of these tumors individually to help us in obviously treatments or looking for treatments. And so the way I like to look at multiomics is this way. So if I'm doing a single omic, what I'm doing is I'm able to see what's happening, and it usually does well. So if we look at TMB, it can help us with PD-L1 treatments, right? We can give us an idea of how the patient's going to respond to it, but it may not be the total picture. It may give us maybe 30% accuracy on those responses. And we're going to talk specifically of an example of PD-L1 shortly. But what if we add another ohm to it? Maybe we look at transcriptomics, maybe we look at proteins. That helps us take a step back and get a grander picture of what we're looking at. And maybe that gives us a better idea of what's actually happening and going on within our tumor type. So that's what we're trying to do with multiomics, and hopefully gives you an ideas when you leave here today of things you might want to do or try to add to some of your studies to get a better, broader picture. So the why right here is kind of patient stratification. Obviously, the more that we know, the more that we can treat our patients and stratify specific cancers into different tumor types. HER2 is a oncogene that's expressed roughly in 1/4 of breast cancers. And when overexpressed, contributes to high rates of cancer cell proliferation. And by knowing this, researchers have been able to develop anti-HER2 therapies, which block that proliferative signal and therefore slow or stop cancer growth. So we can do this with many different genes, many different cancer types, as long as we understand the disease. And then we can stratify the patients based off of therapies that are available by the genomics that we'll talk about. And so what's that lead to? It leads to overall survival rates in cancer. Now, obviously, sequencing isn't really necessarily what's doing that, but it's contributing. Obviously, we're creating more different drugs that can target specific mechanisms in the biology to treat. And some of those can be not specific to a single cancer, but the mechanisms can be found in different cancers, so they can treat different types of cancers as well. So we see over here on the left-hand side, overall median rates of survival from 1970 to 77. And then again in 2007, 2013. And in almost all of these, you can see an increase in the survival rate by all the things that are happening in the studies that are going on in the cancer research realm. Now, in this example right here, we're kind of talking about what used to be done versus what is done now. So before, the way we used to look at DNA, before there was next-gen sequencing, is we used to do a lot of PCR, a lot of Sanger-seq, qPCR for RNA, and Western blotting. And so those were really specific. You had to develop primers and you looked at small areas of the genome. But now with next-gen sequencing, we can do these larger projects. And one of the first ones to do that was the Cancer Genome Atlas, where they were able to profile DNAs and RNAs in cancer, and they could do it on a broad scale. You can do whole exome sequencing, whole genome sequencing to try to better understand these tumor types that we're looking at. And so that's what the power of the sequencing is going to allow you to do, is the scale of your project and the scale of what you're looking at. And so what that allows for, again, is an untargeted and unbiased approach. So we can just go in and look at everything, and then we can define later on maybe what we want to look at. So if we do a whole genome approach and that we find that there's specific genomic markers that are present in the tumor type that we're looking at, then we can develop a targeted panel, and then we can just specifically use that targeted panel, find out our patient stratification based off of those specific genes, or transcripts, or proteins, any of the sort that we can do. And so this is just kind of showing over the past couple of years. Up here on the top part, you see the year-over-year increase in publications using multiomics in 63%. And you can see that that's just growing, and that's going to continue to grow as the assays become easier to perform and the cost of the sequencing goes down. And then down here, you can see the overall increase in grants for multiomic studies. That increases as well. So obviously, going for grants, we want to be able to get more of those. So more grants are being awarded because of multiomics in their studies. So the first example we want to look at is multiomics predicts melanoma treatment response. And so in this one, we're looking at the multiomics from the Long Lab in Australia, and they're looking at PD-L1 therapies. And so traditionally, a lot of people would just look at TMB for this, but this group said, let's go a bit further. Let's try to stratify this a little bit more for our patients and see what specifically, how we can define this treatment option and how they are expressed in the cancer types that we're specifically. And so the first thing they looked at was genomics. And you can see over here, they looked at genomic characteristics, and they did two things. They looked at TMB, tumor mutational burden, so the number of mutations in the exonic regions of an exome, or you can do whole genome just looking at the exons. And they also looked at structural variants. And what they found was with TMB, the higher, if you have TMB high, which is the amount of mutations in a given area, if that was high, then the patients showed good response to the PD-L1 treatment. If they had low, then they showed poor reaction to the treatment with PD-L1. And then with structural variations, they saw the opposite. So if they saw a lot of structural variations within the samples or the patients, then they saw a poor reaction, and then the opposite, a good reaction for low structural variation. Now they want to take that a step further, and they did transcriptomics. So they did full transcriptomics on bulk RNA-seq, and they did some single RNA-seq. And what they found specifically is one pathway here, the interferon response pathway. And what they saw was, as you can see up here, that the interferon pathway was upregulated and good responder. So not only did they have a DNA approach, but they also had an RNA approach that could refine it as well. And then they went one step further, and they did it with epigenomics. So Illumina has what's called an epic array. So you can do methyl sequencing, a targeted methyl sequencing, or whole genome methyl sequencing, but we also have arrays that can obviously do RNA, or they can do methylation. So they use the epic methyl array, and what they found was one specific marker that tied back to the patients. You can see that this gene PSMBU8, and with the poor prognosis, these individuals had a high methylation in those areas, and the people that had low methylation in those specific areas responded better to the PD-L1 treatments. Now, when the group tied all this information together, what they are able to do is with an 89% sensitivity, predict the response rate of people's responses to PD-L1. And so that's what we're trying to do, is trying to understand not only the biology that's happening, but also the treatment options that are available to our patients to make sure that they're the right treatment and that we're going to get the best responses out of those patients. So not only having a single approach to it, but having a broader approach can be more exacting on how the patients are going to react to that treatment specifically. And so the next example we want to look at is multiomics reveals prognosis, therapeutics, and diagnostic insights in meningiomas. And so Dr. Zeta's lab at the University of Toronto, her group used multiomics to investigate meningineomas. And so first, the researchers performed genetic and epigenomic profiling on patient tumor samples. And so you can see here that they were able to divide up in 4 different groups based off of the genomics, and even with the genomic profiles, we can look at the survival rate, and we were able to differentiate the survival rate. So the survival rate up top for 1 in red, and then at the bottom in kind of orange gold for the 4th group. So they took that information and they actually did some RNA sequencing on each of the individual groups to find out the different transcriptional profiles between the different groups that they identified for the meningiomas. And so here in the middle, what you can see is they did this on specifically group 4, the lowest survival rate group down here. And they found 2 different pathways that were unique to that group. One was the HDAC and one was the DDR pathway. And each one of those have a drug that's already associated with it, with treatment for that specific pathway. And so what they did is they took tumors from those people and they introduced them into a mouse model. And then they treated the mouse model with the verinostat for the HDAC pathway. And you can see down here that the verinostat treated mice had longer timelines of survival than the ones that weren't treated. So they were able to take a drug on the market and apply that to patients with that sample to look and hopefully have an overall longer survival rate. Now, I'm not sure if they're into where they are into the study, but kind of gives you an idea of what you can do with finding out how to stratify your different patients, and then some of the treatment options that might be specific to those patients that we can look at. And so the last thing you're able to do is look at proteomics. And with the proteomics, they were actually able to differentiate the different subgroups that they have over here. So you can see here, the subgroup 1, they saw the specific protein marker in that group, the tumor type. And then here for group 2, group 3, and so on. And so you can imagine now, instead of having to do all 3 of these specific assays of DNA-seq, epigenomics, RNA-seq, what if we could just use IHC on the tumor type, identify the proteins that we're seeing, and then do the treatment based off of that on the back end. So using multiomics to better understand the disease, better stratify our patients that we're specifically looking at. And so the final example that I have before moving on to cfRNA is going to be an example of using biomarkers and druggable targets for identification. So in this study, the author took a multiomic approach to investigate several layers of gastric cancer regulation all in one experiment. And so what they're trying to do here is use a multiomic approach to find therapy response characterization and druggable target identification. And so they did it through transcriptomics, genomics, epigenomics, and specifically in the epigenomics ChIP-seq. So the first thing they did is they did the transcriptomics. And what that allowed them to do is divide this gastric cancers into two different groups. You have the EMT and the non-EMT group. So the epithelial to mesenchymal transition pathway group and the non-group. And they did this with 500 genes. So they have two transcriptional profiles based off of those 500 genes that differentiate the two groups. So then they took those two groups and they did DNA sequencing on them, and they looked for specific genes that differentiated the two groups and maybe had different pathways associated with the groups. And so over here on the left-hand side, we can see that over here in red, FGFR2, MET, and then the CDKN2A, CDKN2B were two different or four different genes that differentiated the two groups. And the last thing was they used epigenomics, and they specifically looked for super enhancers. And what they found is with the EMT group, there were 53 unique super enhancers, with the non-EMT and 7, and then we had about 25 overlapping for that. Now, what that allowed them to do was, again, take these different groups, look for drugs that might be on the market specific to specific to those groups, and use that to treat the different groups. And so you can see over here the EMT group with the MET inhibitors and TEAD inhibitors, and the non-EMT with the FGFR, ALK, and EGFR inhibitors. And so they took this and they put it into a mouse model. And you can see the inhibitors based off of the different cancer types, the MOC versus the treatment for the EMLK, ALK, fusion, the MET, and the FGFR2. And we can see specific treatments with those inhibitors and the decrease in the gastric tumor. And so next and last, I want to move on to advances in cancer biomarker research and liquid biopsy. So now we're going to move on to liquid biopsy and how that plays a part in cancer research as well. So one of the things I talked about previously is that not all tumors are physically accessible. So if they're not physically accessible, we need to find another way to be able to identify those tumors and to be able to identify markers for those tumors specifically. And then many patients cannot tolerate tumor biopsy, and sometimes tumors aren't large enough to necessarily get all the tumor. And then as well, many cancers are detected at a late stage where there's not really a great chance of prognosis as well. And so the way we're going to do that is using liquid biopsy instead of tissue. Now, with tissue biopsy, technically, we have a lot of cells there. It's easier to kind of identify the tumor markers. So usually we're looking at variant allele frequencies, so mutation profiles that are in the 5% range when we're looking at solid tumors. When we're looking at liquid biopsy, because we're looking at shedded tumors and tumor cells that are going to be less prevalent in the bloodstream, we need to look down to 0.5 and below percentage variant allele frequencies. So that we have to be able to see at very low frequencies these mutations that are happening, because we're just looking for free-floating either RNA or free-floating DNA in the circulatory system. And so this example is just kind of giving you an idea of the circulatory system and what's kind of going around in there. Specifically, what we want to look at is we're interested in the exosomes that shed cfRNA, circulating tumor cells, and then the cfRNA and the cfDNA that's going to be in the bloodstream. And what some of these different markers are going to allow us to do are look at the genome, epigenome, and transcriptome profiles, and the protein profiles, and that's going to have cancer-associated mutations. It's going to allow us to understand the tissue of origin specifically, and cancer subtypes and different things that are happening within the blood. So we can tie them back to the different areas that the cancers are probably happening and what specific cancers might be present in the patients that we're looking at. And so here we see why is it important for early diagnosis. So there's a lot of companies coming out there. Illumina purchased one called Grail that's looking for early diagnosis in patients with cancer. And you can see there's a reason why we're trying to do this. 9 in 10 patients with early ovarian cancer diagnosis survive for over 5 years, but those that are caught late, 1 in 10 of those people live longer than 5 years. So it's really important to catch these tumors sooner. So liquid biopsy is one way of going and doing this. And so here we're just kind of showing some of the things that we can do with cfRNA. We can look at the cfRNA, cfDNA. We can look at epigenomics. We can look at the transcriptomics from different cell types on here. So we see heart, lung, stomach. We can all identify the different transcriptome profiles that have already been previously established for those specific organs. And so here we're just seeing DNA is identical in every cell except the mutations, and then RNA is dynamic between the cells. So that's how it's easier for us to tell what's happening. Is if we look at cfRNA, we can see specifically where those organs or those areas of the body where those transcriptomic profiles might be happening from, rather than the DNA where it's a little bit tougher to tell because everything's going to be the same in every cell. And so here we're looking at specifically what do we know about cfRNAs in cancer. It's not too much. So this is, again, kind of a very new look at cancer research. So cfRNA is relatively stable, isolated from blood and plasma. So we can store it in Schreck tubes up to 12 months. It's already a little bit degraded when it's in the bloodstream, so we don't have to worry about it degrading as a whole lot after the fact. Versus within fresh and frozen tissue, we worry about it kind of degrading pretty quickly when needed to kind of use it up pretty quickly. And then exosomal release of cancer. So cfRNA is released by exosomes, as I said before. And then cfDNA or RNA markers can be detected in individuals with cancer. And so with that, what we want to try to do is do that sequencing. The way that Illumina would suggest doing it is with an exome approach on the RNA. So we can do a total Illumina RNA prep with enrichment, where the probe sets for the exonic regions. And so you would do your your blood collection using, say, Schreck tubes. We would do an extraction, cfRNA extraction with QIAGEN, like RNA-Easy or one of these kits up here, through our library prep and our sequencing. Now, again, your local core has excellent sequencers. They have a NovaSeq 6000, which is one of the most cost-effective sequencers out there, and it takes a lot of sequencing to do this. So Illumina has a cfDNA RNA or cfDNA assay out there that does about 500 genes, and it takes the equivalent of a 30x whole genome, so 120 gigabases of data to be able to sequence that, because you need a lot of data to get down to those 0.5% to see these mutations that are happening. And the same thing happens with the RNA, where you have to do deep sequencing to be able to identify those different transcripts and the mutations that are happening on those transcripts. And so with that, one thing, I want to thank you all for coming today and talking to us about some of the different ways that we can look at cancer research. I hope that we learned something, and maybe we can take a step back and maybe apply multiomics to some of the cancer research that you all may be doing. But I want to highlight that you have a great local core here. I would hope and I assume that most of you use that local core, but if you don't, I just want to highlight some of the reasons that it's really a good idea to do that. Number one, you're supporting local jobs and local business. The second thing, that you can get help planning some of your experiments, where if you do a send-out lab, all they're going to do is sequence your RNA or your DNA, and then those send-out labs aren't going to test your DNA or RNA. What David and their lab's going to do is they're going to look at your DNA or RNA, and they're going to say, this is good to run on a sequencer, or this is bad to run on a sequencer. And with that, they can save you money. And other things are turnaround times, or if you have grant projects that you need to get in, if you have that established kind of friendship with the core, they may be able to get your samples back into the queue a little quicker, rather than a send out's probably going to put you to the back of the queue. And so with that, this is just some methods, guides that I'll pass this deck out to everybody, but some different decks that talk about different things that I talked about today that you can look over. But I want to thank you again and for coming out, joining us. And if you have any questions, I'm happy to take those. And otherwise, have a wonderful day. I appreciate it. And I'll stick around if anybody has any questions that they just want to ask personally. We'll stick around for another half hour, but thank you again. Yeah. Who can I get those, right? I will. David, are you able to pass the slides out, or is Chris? Okay.